The American Society of Transplantation is proud to present the latest installment of the AST Journal Club series. Today's session features a Journal of the American Society of Nephrology article, Blocking CCL8-CCR8-Mediated Early Allograft Inflammation Improves Kidney Transplant Function. This session is hosted by AST's community of transplant scientists. In a moment, I will turn the session over to our moderator, Dr. Andrew Wells from the University of Pennsylvania. Before we begin the main presentation, we do, however, have a few housekeeping notes to help you engage with today's session. There is currently a viewership polling question displayed for the audience. Please take a moment to answer this question while we finish the remaining announcements. This journal club is being recorded, and the archive will be available on the MyAST website in 2 to 3 business days after the live session. Please note that all of your lines have been muted so that only the presenters can be heard clearly for the archived recording. If you have any question for our panelists during the Journal Club, we encourage you to participate using the Q&A button in the Zoom webinar panel. Questions submitted via the chat window may be missed during the presentation. If there are questions we do not have time for, we will either answer them individually offline, or we will post the full question with the answer on the website following the Journal Club. Finally, when you log off at the conclusion of today's Journal Club, you will see a link to a short evaluation survey to complete. Please fill out this survey to help us keep our content current and engaging. And now, I will turn the session over to our moderator, Dr. Wells, to begin our presentation.
Thanks, Brian. Welcome, everybody. We're fortunate to have Dr. Xunrong Luo with us today from the Duke University School of Medicine to give an interesting recent study on the CCL8-CCR8 axis and macrophages in kidney transplant function. As Brian said, please put your questions in the Q&A panel. I will be monitoring that. And at the end of Dr. Luo's talk, I will draw from those questions in the Q&A for our Q&A session. So with no further ado, Xunrong, please take over.
Thank you, Dr. Wells. So before I start, I just want to preface by saying that the first author of this paper recently unexpectedly moved on from my lab. So I'm stepping in for him because we had committed to presenting this article for the AST Journal Club. So I tried my best to get all the details, information for this paper as much as I can, but I apologize ahead of time if there are certain details that I'm still missing. So the paper that I'm going to present today is titled Blocking CCL8-CCR8-Mediated Early Allograft Inflammation Improves Kidney Transplant Function. I have nothing to disclose. So the aim of the current study is twofold. One is to determine the role of CCL8-CCR8 axis in mediating early kidney allograft inflammation in kidney transplantation. And the second part is to determine the role of donor-resident macrophages in promoting the CCL8-CCR8 axis in early allograft inflammation in kidney transplantation. The model that we used for this paper is a murine model of allogeneic kidney transplantation from a biopsy donor to a C57 Black 6 recipient. In some cases, we had used congenic markers to mark the donor and the recipients for easy identification of donor and recipient cells. So in those cases, we use CD45.2 biopsy and CD45.1 B6. So the background of the study actually started with one of our earlier paper where we looked at single cell transcriptomics of rejecting versus protected kidneys in a mouse model of allogeneic kidney transplantation. And in that paper, we found basically in comparison with rejecting to non-rejecting kidneys, we found two populations of macrophages that expressed a really high level of CCL8. And this got us interested in looking at the role of CCL8, particularly in the macrophage population, in their roles in promoting transplant rejection versus transplant tolerance. So when we looked at the detailed expression of CCL8 in our kidney allografts, we mainly saw that CCL8 expression aggregated in the two macrophage populations that I had pointed out earlier, in that 12 o'clock aggregates of clusters of macrophages. And you can see on the right-hand side of the violin plots, those are the two clusters of macrophages where CCL8 expression was most prominently seen. And its expression is also seen in some other immune cells and kidney parenchyma cells, but comparing to its expression in the two macrophage populations, those were much less prominent. So talking about kidney macrophages, we first wanted to make sure that we know what we were looking at. So we looked at the untransplanted native bulb C kidneys and looked at the different kidney macrophage populations. So what I'm showing you here is a gating strategy of how we looked at these cell populations. We first took the kidney out, we prepared single cell suspension and gated out the CD45 positive hematopoietic cells, and then put it on the CD11B axis. CD11B site scatter. You could see with the site scatter really high population was those neutrophils that we gated out. So we were looking at the CD11B gate and then put the CD11B gate on CD11C at 480. So there you could see the three really clear populations of the red and the green and the blue. So those are the populations that I just want to spend a little bit time describing so that you know what we were looking at. So the red population is F480 high, and that population, and then if you move to the contour plot on the right, you can see that those are CX3CR1 positive, and that didn't really differentiate between the red and the green. But CCR2 is essentially negative in red and more green and more positive in the green population. And more importantly, the red population is entirely Ly6C negative and MHC high. So those are the cells that we consider to be resident macrophages that were thought to differentiate from the yolk sac during embryogenesis. So those are the CX3CR1 positive, CCR2 negative, Ly6C negative, but MHC class 2 positive cells. Those in contrast to the green population, which are cells that we think circulate through the circulation into the kidney. Those are also CX3CR1 positive, but CCR2 positive, Ly6C really high, and MHC class 2 negative populations. Those are the circulating macrophages that we think. And the blue population, which express a high level of the canonical DC marker CD11C, are what we think dendritic cells. So what I want you to focus on, or what I'm going to focus on today, are basically the red population, which is what we call resident kidney macrophages. So this population, what happens after we transplant the donor kidney into the recipient? So what I'm showing you here is a time course of tracing these donor resident kidney macrophages. Remember, these are F480 high and class 2 really high on the CD11B gate. So when you look at this population, what you could see is that at 12 hours post-transplant, it's there. At day 1, it's there. Day 3, it looks like it's actually even increasing in number, but quickly at day 5, day 8, and day 15, they went through a pretty significant crash of numbers. And this kinetics is plotted on the right-hand side in the time course, where you can see the cell number actually did increase in the first 3 days post-transplant, but then quickly disappeared. And by day 15, they're essentially nowhere to be found. And so we were surprised by this finding to learn that these cells actually could increase in numbers. So the next thing that we did was to look at if these cells express proliferation markers to suggest that they can actually proliferate in situ. So that's the bottom T-SNE plot that I'm showing you. So what you can see there is that on the F480 positive gate, within the CD11B positive gate, some of these cells actually did express pretty high levels of Ki67, which is a proliferation marker. So that pleased us to think that these cells indeed, before they crash, they have the capacity to proliferate significantly. That explained why the cell numbers increased at day 3. So those are donor kidney resident macrophages, what happens to them after the kidney was transplanted into the recipient. So what about the recipient infiltrating cells? So we did the same thing of tracing the recipient, in this case, CD45.1 congenitally marked CD11B cells over the same time course. So again, on the F480 y-axis and the MHC class 2 x-axis, what you could see is that this population actually increased over time in numbers, and by day 5, it sort of reached its peak. And those are the cells that are now expressing high levels of F480 and MHC class 2, suggesting that they're acquiring a phenotype of a resident macrophage. And this population appears to reach peak at day 5, and by day 15, essentially, they were the only macrophages in the kidney that could be found. Recall that the donor macrophages essentially completely disappeared by then. So those are the kinetics of the donor versus the recipient macrophages in the kidney after transplantation. So the first thing that we thought that was, we really wanted to look at how CCL8 expression evolves in the donor and the recipient macrophages following the transplantation. So the same gate of the F480 high, MHC class 2 high cells before transplant, this is a naive biopsy kidney before transplant, we essentially didn't see those cells express CCL8, but after transplant, they appear to have a significant upregulation of their CCL8 expression. So what I'm showing you here, CD45.1, CD45.2 to gauge out who's donor, who's recipient, and then put them on the same F480 class 2 plot. You gauge out the macrophages, and we used intracellular staining to look at the CCL8 expression. So what you could see that both the donor and the recipient macrophages appear to begin to express the CCL8 protein. And so on the very right-hand side in the representative plot, what I'm showing you is the CCL8 MFI. So you could see from day 1, they begin to express a little bit, and day 3 significantly upregulated, and in day 7 even higher. But what's kind of interesting is that there always appears to be a slightly more expression of CCL8 in the donor macrophages in comparison to the recipient macrophages, statistically significant at day 7. So that gave us some clue as to what might be inducing who to express what. So CCL8 is a chemokine, and its receptor is CCR8. So the next thing we wanted to look at was who expresses CCR8 in response to the upregulated CCL8 in the kidney allograft. So what I'm showing you here is a feature plot about single cell data where you could see is CCR8 is significantly aggregated in the T cell clusters down at the 7 o'clock area. So we know T cells are expressing CCR8 in our model. So then we looked at who, in terms of the different T cell populations, the CD4 T cells, the CD8 T cells, and the non-CD4 CD8 gamma delta T cells, how much they express the CCR8 receptor that can respond to the CCL8. So this is day 3 post-transplant. We again took the kidney and we looked at cell surface expression of CCR8. You could see that both CD4 and CD8 T cells elaborate this receptor, and so did the gamma delta T cells. And I think what's also interesting to point out is that CD11B cells also express some degree of CCR8, although comparing to the T cells, it appears to be at a slightly lower level. So this CCR8 receptor, which can respond to the CCL8, and that's in mice, appears to be expressed on multiple inflammatory cell populations post-transplant. So then we reasoned, if that is the case, if we block the CCL8 and CCR8 access with either a neutralizing antibody against CCL8 or a blocking antibody against CCR8, we really could sever this access and prevent graft infiltration of immune cells. So that's what we experimented in this experiment. We used CCL8 antibody or anti-CCR8 antibody, both in our model, and treated the recipient for 4 days, from day -1 to day +2. And then we sacrificed the animal at day 3 to look at the kidney allograft infiltrating cells. So what you can see here is indeed, with either anti-CCL8 or CCR8, we were able to significantly reduce CD4 T cell, CD8 T cell infiltration. And it also appears that with CCL8 antibody treatment, we could reduce the myeloid cell infiltration into the allograft as well. But more interestingly to us is that we saw a significant downregulation or reduction of infiltration of these gamma delta T cells that expresses IL-17. So that has not been described before, and it's pretty interesting to see by blocking this access. So if we could block the inflammatory cell infiltration at this early time point, it must be that we could then preserve kidney transplant allograft function in a sort of a longer term. So then we extrapolated our treatment from a short course of 4 days treatment all the way to day 27, and looked at I call it long-term, day 28, longer-term kidney allograft function in this model. And indeed, that you could see that with this treatment regimen, we saw a significant reduction of CD8 T cells. And when we looked at the histology of these kidney allografts, we saw significant less fibrosis in the lower panels, and the creatinine appears to be better. Now, for those of you who measures mouse creatinine, you probably would say that these are very low levels of creatinine, but indeed, in mouse models, the creatinine actually is difficult to measure, and they tended to be very low, and there's the significant difference is usually harder to tease out unless with higher numbers. So we were actually pretty excited to see this degree of lowering of creatinine in this mouse model. But suffice it to say that creatinine is probably not the best measure for allograft function in mouse models of kidney transplant. So that's the blockade of CCL8 and CCR8 story. But then the next thing that we asked ourselves is who actually induces CCL8 to express? And one of the best known inducers for CCL8 expression is interferon gamma. And as we know from the work from the Pittsburgh group, that innate alloimmune recognition frequently induces interferon gamma to be expressed in the allograft without adaptive immunity. So we first asked ourselves, is interferon gamma really upregulated in our system? And the answer is, of course, yes. And so, here we compared our allogeneic transplant to a syngeneic transplant and asked whether or not interferon gamma expression was upregulated. And you can see both at day 3 and day 7, it's upregulated, sort of providing a potential explanation for why CCL8 in our model is expressed. But more definitively, we actually sorted out these kidney macrophages and put them in culture and just stimulated them with or without interferon gamma. So what you can see here is that the macrophages themselves, with exposure to interferon gamma over a course of 2 days significantly upregulated their CCL8 expression. So again, this is sort of more definitive evidence of kidney macrophages under the influence of ambient interferon gamma can upregulate their CCL8 expression. So we think that interferon gamma may be an important upstream inducer for CCL8 in this situation. So that would suggest to us that if the CCL8 is produced by macrophages, and if interferon gamma can be induced in an innate immune recognition sort of situation, if we get rid of the macrophages altogether, then we should salvage the situation by reducing the inflammation. So the next set of experiments is sort of a little bit backwards. We went just upstream to the kidney macrophages that we thought were the main source of CCL8. So in this particular set of experiments, we use clogenate liposomes which is a pretty well-established method to deplete macrophages. We use clogenate liposomes to deplete the macrophages in the donor. It's only in the donor. And then take the kidney out and then transplanted the kidney either with macrophage depletion or without macrophage depletion into the recipient. So the picture that I'm showing, the figure that I'm showing you on the right is basically to show you that interestingly, the clogenate liposome depletion of the donor actually pretty effectively depleted this kidney resident macrophage population. So what you can, what I'm showing you here is with this 7-day treatment of clogenate liposome, by the 8th day, you could see that this F480 high, class 2 high population is essentially gone. But what's more important to us is because we didn't want to take the kidney and transplant them right away under the influence of liposome. So we waited another 6 days. So from day 8 to day 14. And at that point, we took the kidney out and transplanted the kidney then. But what's reassuring to know is that even if we waited for those 6 days, we didn't see this resident macrophage population to return to the kidney, suggesting, I guess, again, that this is an embryonically derived population. So we were happy with that, that we could wait this many days after clogenate treatment of the donor to use the kidney. And panel C is just to show you that the F480 staining in the kidney was essentially completely gone by this treatment, even 6 days following the cessation of the clogenate treatment. So when we used donors whose kidney has been depleted of the resident macrophages, when we transplanted those kidneys, we saw a significant reduction of CCL8 production. So that's the clogenate in comparison to the control PBS liposomes. We saw significant reduction of CCL8 production in this allo transplantation. Of course, CCL8 was nowhere to be found if we only did a syngeneic transplant. So this is an allogeneic specific cytokine that can be reduced by depletion of donor macrophages. And when we did that, we saw that with donor resident macrophage depletion, we saw that there were less infiltration of recipient myeloid cells and less infiltration of recipient CD4 T cells and CD8 T cells. And more interestingly, remember this population of gamma delta T cells that are expressing IL-17 was also significantly reduced by getting rid of the resident macrophages in the kidney of the donor. And this effect persisted to a later time point. This is day 7. We again saw a reduction of the infiltrating recipient macrophages, this F480 positive MHC class 2 positive macrophages, and there's continued to be a significant reduction of IL-17 transcript expression by this treatment. So that these are the cellular phenotype of our kidney transplant when we depleted the donor-resident kidney macrophages. And of course, again, we wanted to see if it translates into kidney function. So we measured creatinine and BUN at day 28 post-transplantation. It looks like we reduced the both creatinine and BUN by depleting the donor macrophages. And this is also correlating with a reduction of kidney injury marker, LCN2. And when we looked at inflammation of the kidneys at day 28, we saw a reduction of intimal arteritis and peritubular capillary arteritis. And mind you here, I wanted to mention that we actually didn't see a difference in anti-donor antibodies, despite the fact that we saw evidence of what appears to be vascular rejection, but we didn't see any evidence of difference of anti-donor antibodies in this depletion strategy. So in summary, we think that this is what's happening during this early stage of kidney transplantation. You have donor kidney macrophages, and they get into the recipient. The innate immune allorecognition causes interferon gamma production, which leads to their production of CCL8, triggering the downstream CCL8-CCR8 axis, which then leads to the recipient monocyte infiltration into the kidney graft and the monocytes to macrophage differentiation. And that led to their production of CCL8, triggering the downstream CCL8-CCR8 axis, leading to recipient T cell infiltration, CD4, CD8, and gamma delta, and graft inflammation, which eventually leads to impaired immediate graft function. So that is what we put together in this paper of what we think could happen early on during kidney transplant that's essentially mediated through donor kidney resident macrophages. So with that, I will stop and thank everybody in my lab who has contributed to this work, as well as our collaborators and funding sources. Thank you.
Thanks, Dr. Luo. So I think, this is a striking result that the donor resident macrophages have this role. So my main questions, and participants feel free to add questions to the chat. To the Q&A. Are really clinical implications. What are your thoughts on using this axis and using your findings clinically? I hopped on clinicaltrials.gov this morning. I couldn't find any CCL8 and MCP2 directed therapies, but there do seem to be some clinical trials on targeting CCR8, and that leads to another question. But is treatment of recipients in the clinic something that you think you could do at this axis?
So to answer your question about CCR8, it's interesting in mouse models. CCL8 and CCR8 is the axis that we know how this chemokine acts through the receptor. In humans, I think the CCL8 receptor is CCR2.
Oh,
so
okay.
we looked at our human kidney biopsy, kidney allograft biopsy, and asked the question of whether or not we can actually see the CCL8 transcript. So we could. And we also looked at CCR2, and again, we could. But these kidney biopsies were taken at all, time frame post-transplant. So I don't really know how, if these findings are significant, but at least we could see that the transcripts were there. Can we target these transcripts in donor or in recipients? Now, donor targeting is kind of difficult because we have very limited ways of targeting donor, and not to mention the ethical issues related to donor targeting. But with the advancement of ex vivo perfusion, with the organs on the perfusion circuit, that especially with prolonged perfusion, there are definitely opportunities that can be used during that limited time period to, think about neutralizing cytokines or blocking certain receptors or depleting certain cell populations. So that's becoming more and more interesting and feasible to look into. Treating recipients, I think, is more feasible, but I think from the mouse studies, again, this will get into which populations are we treating and who are we treating. I don't know. I think that it will be interesting to do and probably more feasible to do. But to me, treating the donors or the circuits will be immediately relevant to our findings.
Yeah, it's exciting. So Dr. Allegre asks, what is the mechanism of fast fibrosis in this model?
So at day 28, so I'm not sure if 28 is considered fast fibrosis, because it's not unusual for people to look at fibrosis in mouse models at day 28. So I think that I wouldn't call this a fast fibrosis model, if that's what she means.
Okay. Megan Levings has asked whether you think this mechanism, these findings, are specific for kidney and kidney resident macrophages, or something you would observe and is ongoing across other organ inflammatory states.
Okay, so I think that's an interesting question. Now, we know that depleting macrophages in various different organ transplantations and tissue transplantations is beneficial. So specifically in islet cell transplantation, as you know that I've been working on islet cell transplantation for quite a while, depleting donor macrophages also has beneficial effect. It's different from glucose homeostasis that we're looking at post-transplantation, but they're beneficial. But whether or not those benefits are mediated through CCL8 and CCR8 interaction, I don't know. I think that macrophages probably do many different things, and the CCL8, CCR8 is just one focus that we zoomed in this paper, but I can imagine that it may do other things as well. So I think that this is why I'm saying that, when we went from CCL8, CCR8 to macrophages, sort of a step back, but it's also a step broader to look at the issues. So I think that it may be effective, but I'm not sure if it's via the CCL8 and CCR8, and that we haven't looked into that. But macrophage depletion we have.
Great, thank you. And Dr. Alessandrini is asking whether you've addressed these questions in sensitized recipients. So if you delete resident macrophages from the donor kidney and put that into a sensitized recipient, presumably with memory T cells, might have the complication of alloantibody there. Have you addressed that, or thoughts on whether this is an axis to target in sensitized recipients?
Yeah, I have not. And I imagine that if memory adaptive immunity is more of a problem in that scenario, targeting this early innate immune process probably wouldn't do a whole lot to a sensitized situation, is my guess, but we haven't really specifically studied this axis using a sensitized model.
Okay. And then another question from Dr. Allegre, just asking again about, in general, if it's not fast fibrosis, what's the mechanism of fibrosis? Do you think it's the macrophages themselves, the gamma delta T cells, the conventional T cells? And then she and I was also going to ask, what about Tregs as well?
Okay, so I think Marissa brought up a good question of how these macrophages may be a contributor to the fibrosis themselves. And we know macrophages kind of, are jack of all trades, do a lot of different things, and they also they change their phenotype through the process of inflammation. Sometimes they are pro-inflammatory, and then over time they may become more reparative. So I think that's a good question. I didn't think about that in terms of how altering the early CCL8-CCR8 interaction could change the recipient macrophages in that regard. So I don't know. It's something very interesting to look into, how their phenotype morph over time if this axis is affected. Thank you.
Great, thank you.
Oh, Tregs.
Yes.
I didn't look at. Sorry.
Yeah. Well, I know I did look into it. I didn't realize there was the species difference between CCR8 and CCR2. I know CCR8 has been implicated on Tregs in the tumor microenvironment, but it sounds like so in the mouse, I do wonder whether if you do have CCR8 expressing Tregs coming into the graft to promote tolerance, is CCR8 targeting pose some problems. But again, it sounds like this isn't going to translate to human.
So CCR8 is expressed on all Tregs at baseline, or this is okay. So because the CCR8,
I think there's a population of highly suppressive tumor Tregs in mouse models that are very high for CCR8. And I think this is the case for human as well. And there are trials in tumor in various solid tumors trying to basically use CCR8 antibodies to deplete Tregs in the tumor.
So our T cells at baseline don't express CCR8,
okay.
but post-transplant, after, I guess they see an allograft, they begin to express CCR8 more prominently.
Great. Well, thank you so much for an interesting and thought-provoking study. If there are no additional questions, one just popped up in the chat. Which I think is what we've already covered. So depletion of donor macrophages does dampen, I guess he's asking the direct alloimmune response. Direct, so question of direct versus indirect. Is that an issue that you have a handle on in this model?
Yeah, so no, we haven't. So specifically, Peter, if you're referring to if we looked at using 4C T cells in this model, which has direct specificity versus something like TCR75 with indirect allo specificity, we haven't specifically looked at that in the CCL8-CCR8 model, but we have looked at how the donor macrophages, possibly through even something like extracellular vesicles and cross-dressing of recipient antigen-presenting cells, can activate 4C cells as well as TCR75 cells in that regard. But in the CCL8-CCR8 model, we did not specifically look at that.
Great, thank you. Well, if there are no further questions, again, thanks so much. This was an interesting Journal Club, and thanks for all the attendees and the participants. Brian, do I turn it over to you?
Yes, thank you so much. AST would like to thank Drs. Luo and Wells and the members of COTS and all of our attendees for today's session. Please remember to complete the evaluation survey in Zoom and visit myast.org/journal club to view our video archives and register for upcoming journal clubs. To learn more about AST's community of transplant scientists, please visit myast.org/COPS or connect directly to the individual COP hub. Thank you again and have a great day.